Hello students, welcome to part 2 of lecture 2 of week 12. So in the first part we looked at the sequencing and assembly methods for metagenomic data construction assembly, and then how to predict genes and what are the common tools. We discussed 3 tools: Prodigal, Gene, Metagenomic and Gleamer, among which Prodigal is commonly used, right, for the gene prediction. So now you have the genes in hand, and down you can go for their annotation and also the construction of MAGs, which is like you can say that partial genomes or kind of recovered genomes from the metagenomes. Okay, so basically annotation of genes is important in metagenomes because in a metagenome there could be multiple bacteria and community basically which is doing a particular function. They are surviving. There could be some species which are playing some role and some the other role. So that system exists and there could be exchange of several substrate and products among themselves. So we have to identify the genes and their functions. So for which one common way is BLAST. You can take the set of predicted genes that you found for a Prodigal and do the BLAST against NCBI-NR database or NCBI bacteria database and see their annotations. But that may be very time consuming, hugely time consuming, and you may like to avoid that. The other method is orthology based using eggNOG-mapper, okay, or you can use interpro-scan, but that may be slow, but it can tell you some additional genes or basically annotations which BLAST or eggNOG-mapper may not found, because it does not look for exact function, but it tells you that partial function or like at least some domain or active site which may be identified, or Ghost Koala, which is a KEGG based tool for annotation. So we will be discussing some of these in this lecture, in this part of the lecture. So one of the common pipeline to do the metagenomic analysis is MG-RAST, which allows you to upload your entire metagenome. Okay, so it will give you option that you can create a login. You can compare against the other metagenomes which are available there. Okay, then you can let it know that what all you want to be analyzed, right? And if you agree that your data becomes publicly free immediately, right, it will do the job faster. But if you keep it private, then it will do this like it will be slightly slower than the usual, right? So this is one option, right? Then eggNOG-mapper we will discuss, and this can be done online, and this can take a lot of data online, which could be good for your annotations, right? Ghost Koala is KEGG based application, right? And interpro-scan, which is motif based or pattern, or basically will give you partial information. Okay, so KEGG is a database of pathways, metabolic pathways, genes, proteins, diseases, reference pathways, right? It also has a very good feature called KEGG orthology, where it has assigned KO numbers to all the genes, okay, so that it can compare a new gene against a known gene and directly assign the same KEGG orthology to that gene, right? And then reference pathways for various metabolic pathways that exist, right? So this makes it a very unique and comprehensive database for pathways. So Ghost Koala is one of the automatic KO assignment KEGG mapping service, where you can upload your sequence or paste it, right? It is available here, and it can assign the KO IDs to your sequences and the pathway IDs also, right? So that is good for like looking at the functional pathways, and so you can try that KEGG application. MG-RAST is a very comprehensive server which can take your data and do multiple things from your data. So it is free to use so far. Okay, so you can register it to run your services and also use the other publicly available pre-computed results to compare. For example, you are dealing with hot springs, so there could be other hot springs available over there, and you can quickly compare the your data with publicly available data sets. Okay, or you can keep your data private and do the analysis, and then do also analysis standalone and then compare, right? So see, basically it has so many available projects, and features that will allow you to do your analysis from start to end. Okay, so this is MG-RAST home basically. So it can do the metabolic analysis, phylogenetic analysis, okay, the functional annotation, the taxonomic analysis, right? And okay, so we will look at it in detail. So basically for the annotation, it is using a seed database it has. Okay, so seed based approach, where like KO or eggNOG, it has also defined or classified the proteins into various functional categories like these, and your data can be classified against these categories, and you will know that which type of functions are more abundant in your data. For the taxonomic or phylogenetic classification, it can compare that like two data set, healthy or diseased, right, and then provide you the classification at the phylum level. And also, like if you ask a question like comparison between firmicutes or bacteroides and like that, which are the commonly present, or the ratio of the two phylums basically has been associated with dysbiosis in several human diseases. Similarly, at the general level or genus level, it can do this same comparison. So basically you can see that okay, these are the different genera, okay, right. So both have these blue, orange and yellow, but the respective proportion varies, which can give you a quick overview of your functional differences between the two samples, like healthy or diseased. Okay, then basically KOGGs and eggNOGs are the common ones which are used. So KOGG is a system of classification developed by NCBI, and Cognita tool was developed as early as the 1990s, which classified the proteins into various categories, functional categories, and that has been improved by using by eggNOG. Okay, so basically that, for example, in this slide you can see that an E. coli classification for different functional categories. So we also discussed this in the previous lecture. So basically G is carbohydrate transport metabolism, E is amino acid transport metabolism. So it gives you a quick overview that what kind of functions or broad class of function is abundant in your data set, and to compare different data sets. So eggNOG-mapper is based on that, the same thing, and basically this classification and the assignment is based on the orthology, right. So it can be used for eukaryotic gene annotation, or prokaryotic, or also metagenomic gene annotation, right. So it has a tool called eggNOG-mapper, which can do from scratch. Also you can even provide metagenomes or genomes or predicted set of proteins. It will use the search engine, which is based on HMMR, Diamond and MMSeq2, right, and compares against its own database that it has, eggNOG database that it has. Then it will calculate the orthology of the protein, okay, using the pre-classified orthology information it has, okay, and then do the function annotation using the against the KOGG or functional categories or PFAM or eggNOG. So it is very comprehensive, and then it will provide you the annotated protein names and the further information and the PFAM domains also. Okay, so this is the pipeline for eggNOG which we just discussed. So it is a very comprehensive tool, and good thing is that it is available online, so you can upload your sequences and you can use it. So you can upload either protein, CDS, genomic data, metagenomic, or even seeds, right, from MG-RAST, okay, output, right, and upload the file and you can get your annotation. Okay, so this is like typical outcome. So your protein name, okay, and the e-value and score of that alignment, okay, then what is the functional description of the protein, what eggNOG ID it matches to, right, the GO classification, the KO classification, the KEGG pathway classification, so which is very good and comprehensive. So in one go you can get all the functional details. Okay, then in the next part we will deal with genome resolved metagenomes. Okay, so basically your last idea, or basically the final aim should be to recover the complete genomes from metagenomes, right. but it may be limited by the amount of sequencing, because you have just fragmented each and every bacteria that was there in their respective copy numbers, and you have lost the information of which fragment is coming from what genome, right? So again, to reconstruct the genomes is a challenge, but at least you can try, and that is the concept behind the metagenomic assembled genomes, where you will bin the similar looking or basically the reads which are coming from a single bacteria or species possibly into various MAGs, okay, and then you can do further operations on these MAGs, okay. So basically this concept is very important, and it can be used, has been used recently for several publications, right, and it enables you to identify the novel species, and because some MAGs you may not be able to classify, or that will look like any other known genome. So those could be the new species that you may have found, which you may not have found otherwise, because this way you have also classified or segregated the data, okay, all the okay, there could be some bins which can be easily called for, and they, okay, and some bins which are unique and unclassified. So you can do more with these kind, and don't get confused with like too much or bulk of the data all the time, okay. So like processing will be like you will have this sequence read. Once you have done the quality filtering and other steps, you will go for the de novo assembly, okay, and then basically the contigs that you have got, you can classify them into various bins, right, and using several criteria like composition or marker genes that we will discuss later, right, and then do the gene prediction in those and the annotation, and then try to reconstruct the genome. This is the assembly based approach, okay, MAG approach. And reference based, that you may want to do like if you have sufficient data, is like you map your reads against a reference database where you know that these are the genomes, right, and you can map the reads against those genome. Like E. coli, you map all the data against that E. coli genome, and some reads will match to that, and then that will help you to reconstruct E. coli like genomes from using the reference based alignment approach, okay. So this is another way, but MAGs helps you to identify new genomes, whereas reference based approach can help you to quickly re-identify those genomes. So the MAGs can be done on the basis of tetranucleotide frequency, which you can calculate, that can like roughly they can segregate the different bins, because they should be very different in terms of tetranucleotide frequency, which is a kind of feature of different bacterial genomes. Very close bacteria or groups of bacteria like species level, okay, they may have some issues with or limitations with using tetranucleotide frequency. Or the presence of marker genes is also very helpful. This concept is gaining a lot of importance these days. Alignment against taxonomic groups or known data, okay. Codon usage and these kind of features can also be used for the MAG clustering and classification. So basically idea is to represent the whole genome of an organism, which you have recovered from the metagenomes contigs, right. So basically several tools are there for construction of MAGs and their annotation, like MetaBat2, Concat, or this MetaRap. So we will be looking at these tools in slight detail. So Concoct is one of the tool that has come in nature methods, and it is based on reconstructing the genomes from the metagenomic data, okay. So this is also widely used, and they have also demonstrated on the human gut microbiome data, okay. MetaBat2 is another tool that is used to quickly do the binning and reconstruction of metagenomic assemblies from the contigs or the directly from the reads, right. So basically this is also used, and you can download it from this site. MetaRap is one of the commonly used tool because it is a wrapper program, or you can say software which runs several other like Concoct also or MetaBat2 also for together to do this job of basically it can classify them, the raw sequence read into metagenomic bins, and also perform their analysis. It can use multiple other tools and software to do this job in a hybrid manner, right, and will provide you the metagenomic build, taxonomy, abundance information, functional, and also some visualization of your data, right. So this is also a good and commonly used tool. So this is MetaRap pipeline. So it takes raw read, do the quality QC. So basically you can say that it is running from scratch. Raw data can be given. It will do the QC, clean the reads, okay, do the assembly, right, run the assembly module to do the assembly, do the binning module to get your metabat bins, max bins, and cockdown bins. So basically these two programs we have seen earlier also. So it will do assembly using multiple programs, and then do the bin refinement. That means whether the bins or the clusters of contigs or raw reads that you have made is correct or not, okay. So there are some correct criteria that can be used to check whether the quality of mag is correct, good or not, okay. Then make the final bins and then do the quantifications and like what type of genomes are more abundant into heat maps or blob plots or taxonomic information and function annotation of those. So it's a very comprehensive tool, and also basically it runs Kraken, which is a very good and fast classifier for metagenomic reads. Okay, so quality filtration evaluation, that means whether the max or cluster that you have got is complete or not, whether it is, how much complete it is, whether there is a contamination or this contig should have gone there, or there could be one gene which could be present in multiple genomes are highly identical. So you may like to place that contig into multiple of these mags. So all these things can be resolved, okay. So for that, CheckM is one of the tool that can determine the quality of mags, right. So basically ultimately you will get mags classified as finished or high quality or medium quality or low quality, and you may decide that which ones you can keep for further analysis. And like the good quality should have at least 90% completeness. That means it is covering a respective genome almost 90% and has less than 5% contamination from other mags which you have also found during the analysis. Okay, so basically it's a very fast tool that has a lot of data from other bacterial and marker and taxonomic databases, okay. And basically it also has a bin annotation tool, BAT, that can perform the taxonomic classification of mags by using the open reading frames and from each map, and then mapping it against the NCBI-NR protein database, right. And phylophylline is a tool for the phylogenetic classification of the mags, because you have got the mags which are representative genomes or basically putative genomes or partial recovered genomes from the metagenome, and you may like to put them and you have also seen that they could be very good quality, low quality, poor quality, finished or complete, like that. So you would like to put that in form of the phylogenetic tree, which can be done by phylophylline using the marker based approach, right. So the contig annotation tool and bin annotation tools are also available at the GitHub along with the MetaRap, okay. So this is the phylophylline, okay, contig annotation to cat and bin annotation BAT. So cat, BAT and phylophylline are also available for the annotation and also the taxonomic classification and the phylogenetic classification of mags, right. There is one more pipeline, Squeeze Meta, which is a fully automated metagenomic pipeline. So I am just giving you some information that what other things are present, and the best way to, when you start your analysis, is to go for a literature search and see that what is the new tool that has come. Most of these will be you may find that you may get a newer version of the tool that we have discussed, or a new tool that might have compared itself with the existing or earlier gold standard tool, and you may like to use that tool for your analysis. Okay, then when you have got so basically you can do metagenomic analysis using the metagenomic data, okay, which is the shotgun data from the actual genomes, but you may also do something similar using the taxonomic reads also, where you use the 16S ribosomal RNA marker based approach, or the V3 region or V4 region or combined region, okay, at least some marker region to calculate the abundance and taxonomy, right? So PICRUSt can use that data to basically, because 116S ribosomal RNA, when I know it is coming from this genome, I know that what genes that genome contains, so I can use that information from all the genome that I have predicted using the taxonomic prediction that I have done, to retrieve their gene content, okay, and then doing the functional analysis. So PICRUSt is one of the way of quickly assigning functional annotation to the, or making functional predictions from your taxonomic data. So it's a very useful tool, and people like to, even those who only the marker based analysis may use PICRUSt. Now it is 2 version for their quick functional annotation of the metagenomic data. And once the amount of data also increases, right, like that what genomes are there, and you have a very good information about the markers from those genomes, even 16S ribosomal analysis should be sufficient, because it will be cheaper also, and you followed by something like PICRUSt approach, which can be used to use the taxonomic information to retrieve the functional information from the genome that already exists. So you can download it from GitHub and install and use it, okay. So it can quickly assign your OTUs or ASVs into and give their functional annotation, and can also build the maps of those genes and assign them to pathways. And it has been demonstrated very well performance on the gene sets where both taxonomic and metagenomic data was produced, and it match the results for the taxonomic and function analysis, right? So basically it provides KO, EC and pathway abundance as the output, which is relevant for any metagenomic analysis, okay. So at the end, in this lecture, we followed it up with the sequencing and assembly and gene prediction that we did in the last part of this lecture, and we concluded this lecture with the methods that are used for the function annotation of the genes that we predicted: partial genes, complete genes, and what are MAGs, how they are constructed, and how their function annotation is done. So this is good for today. Thank you.